So aging is one of those big problems that Nadim graciously mentioned when he talked about some of these issues that genomics is going to be facing within the next few decades. So when you see these babies, what do you feel? Joy, maybe their ambition, their potential, maybe even nostalgia. Now, what about death? When you imagine death, how do you feel? Sadness, maybe coldness, maybe you think that's inevitable. We as a species, as humans, have been conditioned to think that death and aging are just parts of the human experience, and they are inevitable. They're just parts, like they're as normal as eating, sleeping, and growing up. And of course we believe this way because as a species, we had no other alternative. There was no alternative to dying. You would be born, you would grow up, you'd have children, and then you'd die. And this has been the way since forever. But recently, there's been a big change. We're looking at aging at a completely different way. We're looking at aging not as a reality that we cannot avoid, but rather as a disease which infects 100% of the population and also kills 100% of the population. So naturally, my team and I wanted to help contribute to this fight. So we did what we know best, and we did this with genomics, specifically with epigenetics. So epigenetics involves less of the CRISPR-Cas9, like you guys may have known, and more of what Nadim also expressed, gene expression and repression. So epigenetics deals with the human genome, with the human genome's interaction with the epigenome. So the epigenome is the amalgamation of all these proteins and enzymes in your body, which manipulate the genome in every possible way, except for mutation of the DNA. So any other way you can affect DNA, aside from replacing A, T, C, or G. That is what epigenetics deals with. And the problem that we want to tackle was something called telomeric decay. So what you're looking at here is called the chromosome, the basic DNA structure in a cell. And here, labeled in green, are your telomeres. Now, on the surface, telomeres may not seem like they do much. They're just little caps there, kind of like how on your shoelace, you got a little plastic cap there too. But even though these telomeres do not code for anything, unlike the rest of the chromosome, they're far from useless. As you age, your telomeres decay. And this is not a causation. This is not a correlation. This is a causation of aging. As your telomeres age, we know, as you age, we know that your telomeres decay. And it is now recently a big issue seeing how we can reverse this, how we can stop telomeric decay in ordinary cells. So how do we go about by doing this? Well, how does the cell go about doing this? Well, how it works is that every time your cell divides, a bit of the telomere gets shaved off. And you start with a cell with a lot of telomeres, and then as it divides, a little bit is shaved off. And then it divides again, and a little bit more is shaved off. And this keeps going until your cell reaches what we call a Hayflick limit, or essentially a division limit. And at this point, your cell is left with two choices. One, screw the rules, I'm going to keep dividing and kill myself, basically, or become mutant. And this is a rare occurrence, but it can lead to oftentimes mutagenic diseases like cancer, or it can kill the cell. But something more common happens usually, and the cell obeys by the rules, and it obeys by its limit, and it stops dividing. But it doesn't die. It just stays there in this kind of zombie state. And we call this cellular senescence. And this is also another big correlation of aging. So here the cell does nothing but kind of stay there, annoy its neighbors, and not really do anything. And this, as senescent cells accumulate in the body, what you get are more effects of aging. So this is also related to telomeric decay. And as you can see, the decay of the telomere seems to be kind of reaching everywhere, causing aging throughout the entire body. And this is where we want to stop things. So there are immortal cells out there, despite what you might think. So even though our normal cells are not immortal, cancer cells and stem cells are. And we wondered, what do these cells have in common that makes them immortal? Cancer cells and stem cells on the surface should be very different. One can kill you, one can save you. And the answer lies in one enzyme called telomerase. So telomerase is crucial in rebuilding telomeres as they disintegrate. So every time the cell divides, you lose a little bit of your telomere. But cells with telomerase still happen to have this enzyme, which come in and they rebuild the telomeres. So every time the cell divides and you lose a little bit of your telomere, telomerase comes in and plops it back on. And this essentially this is like a life hack, literally, because you are hacking how you don't die. The cells continue to divide indefinitely. And this is the magic of cancer and stem cells. They can't die because of this reason. So we ask this question, well then, if these cells can have the privilege of not dying, why can't our hard-working skin cells and liver cells have the same privilege? And the answer lies in epigenetics. So telomerase is coded for a gene called H-TERT. And H-TERT is regulated in somatic cells and is essentially repressed to the point it is not even active. And the repression of this gene is done by chemical markers known as methylation groups or methyl groups. And these methyl groups can be plopped on both the DNA, like the gene itself, or it can be placed on like these coiling structures where the gene is coiled around, called a histone. And the accumulation of these marks, oftentimes, with some exceptions, results in the repression of the gene. So our team identified one key methylation mark called H3K9ME3 on the histone where the H-TER gene is. And we figured if we can somehow get rid of this gene or stop this gene from being maintained, then H-TER should be able to be regulated, and maybe we can get more telomerase going. So we identified the enzyme that places this specific mark and maintains it, because epigenetic marks need to be maintained. If they're not maintained, then they have the risk of possibility of fading away. So we figured this out, and we figured out what specific enzyme lays down this mark and maintains it. And we thought, okay, if we block the enzyme that does this, and that lowers down, that means we'll have less of this mark, which means more expression of the H-TER gene, which then means more telomerase. So how do we do this? What inhibitor do we use? What delivery mechanism do we use? We have our solution in here. Pitocin is a drug, is a nonspecific inhibitor, meaning it targets our enzyme unspecifically, and we can deliver it to our targets to stop the enzyme from delivering the H3K9 methylation. And when we do this, when we eliminate the maintenance and the establishment of the mark, we allow the gene to be expressed. But one problem still remains. We have our drug, we have our entire problem here, but we still don't know how to deliver it. And this is a big problem when it comes to pharmaceutical companies and scientists everywhere. How do you effectively deliver a drug? And this is where we also want to take the liberty to solve that problem. So U of T recently came up with a new idea called personalized nanoparticles. And we still haven't got in touch with one of the PhDs who are running this research project, and we got to look at their paper. And essentially, personalized nanoparticles are a new way of delivering drugs, where you take biological fluid or tissues from the patient's own body, and you incubate it in a special way where a new biological chemical shell is formed in place of, let's say, a gold nanoparticle. And this can be used as a drug delivery tool where no immune response in the person's body is generated because the nanoparticle itself came from the person's own body. So combining this with our new drug, we created an overall treatment model to solve cellular immortality. Now, there are a few questions that we still need to answer, like how are we going to actually navigate this new combination of drug and delivery mechanism towards the nucleus and really get accurate targeting. So we still have some questions to answer. But should it be us or another group that answers these questions? What we may be onto is a new way of treating aging at the cellular level, and to possibly create immortal tissues in the future that don't wither with age as we may expect now. So going back to kind of the idea of life and death. What you see here is a grandpa and his daughter, and his granddaughter. So this grandpa was born, and he expected to probably live to around 70. But thanks to his lucky genes and probably modern medicine, he's lived to 90. And had his granddaughter been born in the same era as he, it would have been the same case for her. But when his granddaughter reaches her grandpa's age, she'll be lucky to find out that not only is her skin not wilting away, or her organs are not going short, or her joints are not weakening, but she'll find herself to be, hopefully and ideally, at the same space as she was maybe when she was 30 or 40. So it all starts with a mindset change. Despite the complex science, this is all the products of a small, simplistic mindset change, where we look at aging not as the death specter that kills us all, or something that we cannot control, but rather, instead of having aging control us, we're looking now to control aging itself. Thank you.